Alright, so you've heard of genes, and you've heard of therapy. But what is gene therapy? Well, gene therapy is the introduction of a normal gene into an individual's genome in order to repair a mutation that causes a genetic disease. So basically, it's using genes to help treat or prevent diseases.
Here's a little bit of a history lesson for you guys. Feel free to fall asleep. In 1985, Dr. W. French Anderson and Michael Blees in the National Heart, Lung, and Blood Institute and the National Cancer Institute worked together and showed that the cells from patients with ADA deficiency could be corrected in a lab setting. In 1986, the team studied transferring genes into bone marrow cells of animals. However, the number of cells that actually got to the genes was too small to be affected.
In 1988, the researchers decided to use white blood cells instead of bone marrow cells. This was a very successful experiment, and they wanted to test it on people. They then worked with Dr. Steven Rosenberg to test the safety and effectiveness of gene therapy on cancer patients. Basically, scientists took cells from a tumor and modified it so that when put back into the human body, it would kill the tumors. This confirmed that the virus that they made could be used safely in humans.
Alright, now this is big. In 1990, doctors Anderson, Blaze, and Kluver used a virus to deliver the correct ADA gene into 2 young girls with ADA deficiency. This treatment was ongoing for about 2 years. Now the 2 ADA patients are leading completely normal lives. In 1993, researchers used gene therapy to treat newborn babies with ADA deficiency by delivering the correct genes to the cells from the baby's umbilical cords. The baby showed a steady increase of ADA in their immune cells following just 1 gene therapy treatment.
How does gene therapy actually work? We'll be going more in depth with this a little later on, but here's some general info. Gene therapy is used to introduce genetic material into the cells in order to make certain helpful proteins. It's also used to compensate for other genes that may not be functioning properly. The gene isn't directly inserted into the cell, as it wouldn't carry out the desired effect.
A carrier, called a vector, is used to transfer the gene and is usually a virus that has been modified so it won't cause disease in people. The vector can either be injected or given by IV directly into a certain tissue of the body. The cells with the new gene will then multiply and produce enough of the gene product for the entire body, therefore restoring the undiseased phenotype.
So, in the best case scenario, gene therapy simply adds a functional copy of the gene of interest to cells that only have non-functional copies. However, it's not always as simple. One way of repairing a mutation in a gene is called SMART. SMART stands for Spliceosome-Mediated RNA Transplicing. This procedure repairs the mRNA that's copied from the mutated gene. Also, there have been several viral vectors that have been developed in order to repair mutations that occur in the DNA. This technique uses restriction enzymes to cut out the faulty sequence and replaces it with a functional one.